I'm speaking today with Skirmantas Kriaucionis, who is joining us from his lab at the University of Oxford in the UK. We'll be discussing a paper that Dr. Kriaucionis co-authored and that's being published in the high-profile journal Cell. Senior author on the paper is Nat Hines from Rockefeller University in New York City. Before we jump into the science, Dr. Kriaucionis, I thought it might be helpful to give our listeners a little bit of insight into your background. You did your graduate work in Adrian Bird's lab, which is where you and I met, and then you went on to do your postdoc in Dr. Hines's lab. And
yes.
I suspect you probably didn't expect to publish a paper on MECP2 from the time that you spent in the Hines lab, but that's how things turned out, and in a certain respect, you kind of come full circle. But let's begin with an earlier paper, a seminal paper, really, which was published in Science while you were in the Hines lab, in which you discussed the existence of a new nucleotide base, 5-hydroxymethylcytosine. Can you elaborate on this?
Yes. So this is our 2009 paper in Science, and this was as well a very unexpected discovery. We were looking at chromatin structure differences between different neuronal cell types, and we used a certain methodology which is not being commonly used, and by thorough observation of the results we observed additional signal which later we identified as a novel DNA modification, and this was really unexpected for us as well as for the community, and we invested a lot of time and experiments to prove that it is indeed the case that additional modification exists in the DNA, and this was really well received, and up to now there is over 400 papers published on that topic.
Yes, it's been amazing. It's a really hot area right now that's being looked at. I remember you and I had a conversation probably in 2008, and it might have been early 2008. I remember it was cold outside, where you were telling me about this finding, and you were quite excited about it. So in this paper, in the Cell paper, you identify MECP2 as the primary protein that binds to hydroxymethylcytosine as well as to methylcytosines. Can you elaborate on this and tell me whether you were surprised by this finding?
Yes, we were surprised. We were really interested to find a protein which recognizes 5-hydroxymethylcytosine, and this is based from the previous work which was done by a number of groups and which Adrian Bird actually contributed to show that 5-methylcytosine in the genome is interpreted by readers of 5-methylcytosine, by methylcytosine binding proteins in the genome. Therefore, extrapolating from this work, we thought that a protein might exist which would specifically recognize 5-hydroxymethylcytosine, a novel DNA base which we identified. And this inspired to do experiments and investigations in which we, in unbiased fashion, tried to fish out the protein which would recognize 5-hydroxymethylcytosine. And to my big personal surprise, we got MECP2 as a binding partner, which binds to 5-hydroxymethylcytosine as well as 5-methylcytosine. And this was completely unbiased thing, and it was really surprise for me to see the results coming from the screen, which showed that this is MECP2, which showed such a convincing binding to 5-hydroxymethylcytosine.
Yeah, I think it's important to point out to parents or non-scientists who may be listening to the podcast the importance of the fact that this was unbiased. It's not that you set out with a hypothesis that you thought MECP2 would be the primary binding protein and then set out to find data that would support your hypothesis, but rather you said we don't know what it is and we're going to try to find out what it is.
Yes, indeed. I think this approach, unbiased approach, always adds additional value to the investigation and really allows to examine the whole spectrum of possibilities rather than focusing on one specific.
So can you tell our listeners from your perspective, what are the key highlights of the Cell paper?
So the key highlights are, first of all, in addition to identifying MECP2 as a binder for 5-hydroxymethylcytosine, we as well invested a lot of effort to look where 5-hydroxymethylcytosine is localizing in different neuronal cell types which are obtained from in vivo tissues. And we used 3 different cell types from cerebellum, and we mapped 5-hydroxymethylcytosine in the genome, and we found that it localizes on the transcribed gene body. So it localizes on active genes, basically. And the second part of the story is that we identified protein MECP2 as 5-hydroxymethylcytosine binder, and this was really an interesting connection to find the protein which binds 5-methylcytosine and 5-hydroxymethylcytosine and localizes both in the repressed heterochromatic regions and both on actively transcribed genes. So this was a relation between these 2 observations. First of all, understanding where 5-hydroxymethylcytosine in the genome is localized, and second, what is the protein which reads 5-hydroxymethylcytosine signal. And the third, I think, very important point was to really understand whether or not how MECP2 is actually interpreting 5-hydroxymethylcytosine and 5-methylcytosine signal, and whether or not it's relevant to Rett syndrome. We found a mutation, Rett syndrome causing mutation, R133C mutation, which predominantly abolishes recognition of 5-hydroxymethylcytosine but still has affinity to 5-methylcytosine. And this mutation is really interesting because it allows us to speculate that MECP2 binding to 5-hydroxymethylcytosine is important, and it is important as a part of the function which causes Rett syndrome.
The 133C mutation is considered to be one of the more milder mutations in Rett syndrome. Does
yes,
that
that's
have
right.
any relevance to your finding?
Yes, that's right, and I think it's really interesting because R133 mutation has been characterized by delayed onset of regression and with somehow improved speech and motor skills, and therefore we think that it's not complete loss of function. So binding of MECP2 to 5-methylcytosine is still a very important part of the function for MECP2, but if we are able to abolish 5-hydroxymethylcytosine, which as well plays a function in Rett syndrome, we get not a completely classical Rett syndrome, but a slightly milder version of it. So we don't exactly know at the moment